welcome to NPTEL online course on Introduction to Complex Biological System. Now we are on module 2. We are discussing about transcription and translation, making product from the stored information in DNA. So this is lecture 10 and we will be discussing about regulation of gene expression. So here mostly I will cover few important points. Why is regulation of gene expression important? That is the first point, followed by an overview of regulation of gene expression, and finally I will give you one classic example of gene regulation mechanism in bacteria, that is also known as lac operon system. So here first, why is regulation of gene expression important? So if you see, each cell contains all the genetic material for growth and development. But the thing is, some of these genes are expressed all the time and other genes are not expressed all the time. They are switched on and off at need. For example, if I now say about human body, every cell they have same information, whatever written in DNA. But now if I compare a nerve cell and a liver cell, those two cells are completely different, how they look like as well as their function. So now why this different behavior, different functions are actually coming? So all those things are actually lies how the genes are getting expressed in these two different types of cells, and that actually determine the fate of the cells. So now if I give some example, like here one protein, if I say GAP-DH, this protein is present in every cell in our body. The full form of this protein is glyceraldehyde So glyceraldehyde 3-phosphate dehydrogenase, so GAP-DH. So this helps in glucose metabolism. So as we know, for living cell, each cell they need to metabolize glucose and they need to produce ATP. So this enzyme is present everywhere, and I would say this gene is also getting expressed. That means it is making product from this gene all the time. Like some genes might be expressed, might be making product all the time, but some genes they will only make product at need. And apart from that, we can also classify this kind of genes in in two defined way also. One is called constitutive gene expression. That means those genes are constantly expressed or they are available in every cell. Like genes are present in every cell, but they are getting expressed, they are making product in every cell. For example, GAP-DH. But insulin, this insulin gene is tissue specific gene because insulin is getting produced in pancreas only. So as a result of that, although the insulin gene is present in DNA, present in every cell, but it is making the product, it is getting expressed inside pancreas so that pancreas can make insulin for us. So this is called tissue specific gene expression. So like the summary of this slide is that gene expression is very much controlled. So although we have the same set of information in every cell, but because of gene expression and its regulation, we see different types of complexities and different types of function. This is not only in eukaryote, even this is true for bacterial system. For example, a bacterium when present in nature, and that bacteria might be pathogenic also, but when it enter inside host, then for survival it can make different types of product, different set of genes will be expressed so that it can actually evade host immune system. So as a result of that, this gene expression and its regulation is true everywhere in prokaryote as well as in eukaryotic cell. Now, I will just give you an overview of gene expression. So, gene expression means to make product from the gene, right? So, if I say again just like central dogma, so DNA goes to RNA. So, that is transcription. And here particularly I am writing messenger RNA, so that it is the direct template for translation and it will make a product, that is our protein. This is translation. Now, major regulation actually restricted to this part, the transcription and translation. Like if we can control the regulation of transcription and translation, then the product formation will be, will differ. So, for example, transcriptional control, if I give some example, RNA polymerase transcribe DNA or particular gene to make RNA. Now, if the promoter sequence which is present just before the gene sequence, if the promoter is very strong, that means it can recruit RNA polymerase very efficiently, then the amount of RNA or the transcription will be higher. So, as a result of that, more mRNA will be present inside the cell, and as a result of that, if the more template for protein production is there, so naturally we might get more protein. But that is not always true, because there are so many other type of regulation mechanism exists. Now, this mRNA can form protein that already I mentioned, but there is specific mechanism that is called the degradation of mRNA, because mRNA if exists all the time inside the cell, then it will constantly make protein. But I just told that sometime we don't need that protein. So, as a result of that, mRNA should be degraded also. So, that is degradation of mRNA. And then protein is getting produced after translation. But this protein, there are many fate of this protein. Although protein is present, this protein may be active or inactive also, because finally protein will do function for us. But now, although we have protein, if that protein is inactive, then we are not getting the function. So, there are very well known mechanism present in our body as well as in bacteria. For example, phosphorylation and dephosphorylation of protein, that is a kind of very well known mechanism that regulate protein activity. So, although protein is present, but it depends whether it is active or inactive version of the protein. And then this protein can also be degraded by well known mechanism that protein can be degraded also. So, this is degradation of proteins. Now, this scheme, whatever I mentioned, this is true for bacteria as well as in eukaryotic system. But there are so many complexities present in eukaryotic system, and few of them I will highlight. So, particularly in case of eukaryote, we have nuclear membrane. So, if I say if this is nuclear membrane, so this is nuclear pore, So, this is cytoplasm and this is nucleus. Now, we have DNA here inside the nucleus, DNA first goes to mRNA transcript. Then this is before processing. In eukaryotic system, mRNA should be processed properly in order to make mature mRNA, which is the template for translation. So then after processing, we will get mRNA. So this is processing step, and then this mRNA will be transported into cytoplasm, and this is readily available for translation. So now it will undergo translation and it will make protein. Now specifically I would like to mention this processing step here. So during processing, I mentioned that processing means at the 5' end of mRNA, the addition of 7 mg cap, and at the 3' end poly A tail should be added, and also very importantly there are many introns present in case of eukaryotic system in eukaryotic gene, but those introns should be removed, and finally we will have our mature mRNA without intron. So this processing, this splicing of mRNA, it is also kind of very much controlled, and it can also affect in gene expression. So I will give you just one example here, that how splicing of mRNA actually help in gene regulation, or that can control gene regulation. So here if I mention here, this is 5', 3', this is an mRNA, and this mRNA here, this is exon 1, exon 2 and exon 3, and this is intron I1 and I2, this is intron 1 and intron 2. So now splicing means I told that introns should be removed. So as a result of that, what should I get is 5' to 3', this is mature mRNA, and you have E1, E2 and E3. So now this is your mature mRNA, and it will make some protein. Okay. After translation, it will make a protein. Now sometime for specific genes, alternate splicing can also happen. In that case, what we will get, because of the alternate splicing, so we might get 5' to 3' this mRNA, but here you have just E1 and E2, but the E3 portion is not present in this mRNA. So as a result of that, it will also make a protein. Now there should be some difference in these two proteins. For example, I would say if in this protein, for example, I am saying that protein A1 and protein A2. In protein A1, if I say this is a membrane bound protein, and the E3 portion actually help this protein to be anchored on the membrane of a cell. Now protein A2, they do not have this E3 portion, right? E3, that means the exon 3 portion. So as a result of that, when it will be translated, that portion which will be required for anchoring on the surface of cell membrane, it should not be there. So as a result of that, it will not be present on the cell surface. It can be secreted outside the cell. So this is kind of complex mechanism of gene regulation also, and this kind of different type of regulation or gene expression is present in our system. And this is just one example I mentioned, particularly this is for eukaryotic system, and also this mRNA degradation mechanism, it is also very robust. There are many different types of mRNA degradation mechanism present. For example, I just mentioned in previous classes that 3 different types of RNA, particularly messenger RNA, ribosomal RNA, and transfer RNA. Those are the very important classic RNA, which is very basic thing. But apart from that, we have some small RNA which regulate gene expression. For example, micro RNA. So that means this micro RNA can also regulate the degradation of mRNA. So this microRNA is small RNA, but they can control they can control the mRNA, that how long they will be present inside the cell. Similarly, siRNA. So siRNA, so that is also very important, and that also regulate the mRNA function. So this is microRNA and siRNA, and very recently for microRNA in 2024, Victor Ambros and Gary Rubkin, they got Nobel Prize in Physiology and Medicine for their work on microRNA. And similarly, in 2006, Andrew Fire and Craig Mello, they also received Nobel Prize for their work on siRNA. And so you can understand that how they are important in gene expression, microRNA and siRNA. And then this protein degradation, whatever I told, degradation, and then protein, whether it will be active or inactive, also those are very important in gene expression. So now specifically, I would like to mention a case study. For example, this is in bacterial system, that lac operon. This is a classic example of bacterial gene expression control. Whatever I discussed in the previous, during the last few minutes, that is overall, overall understanding about gene expression. But in this case, I would be specifically mentioning a set of genes, how they are getting regulated in order to metabolize some particular, for example, lactose in this case. So lac operon. So operon means here cluster of genes related by function and regulated by a single promoter and transcribed into one mRNA. This is kind of operon. So this operon actually, this word I believe came from Latin word opera. So that means perform or work. So something like that. So now if you see, this is cluster of genes, as just I mentioned, cluster of genes. So you can see here we are mentioning 3 genes here, LAC-Z, LAC-Y and LAC-A. So this gene LAC-Z, so gene means some sequence present in DNA, and it will make some product after transcription and translation. So LAC-Z, it will make a protein, particularly this is an enzyme called beta galactosidase. This is the product of LAC-Z. Similarly, LAC-A, so LAC-A will produce another enzyme called lactose permease. And the third one, LAC-A, it will make some enzyme, it's a kind of big name, thiogalactoside transacetylase. So that actually help in cell detoxification, but this kind of complex mechanism, we are not going to discuss that part, but still it should be fine. So, this is thiogalactoside transacetylase help in cell detoxification. And beta galactosidase, like many of you know already, that lactose. So, this lac operon we are discussing to understand how bacteria they regulate or they actually metabolize lactose. So, lactose should be metabolized into glucose and galactose. Lactose should be broken down into glucose and galactose because lactose is a disaccharide. So, it should be broken down into glucose and galactose for for production of energy. So, lactose, it will make glucose plus galactose. So, this conversion is done by this enzyme called beta galactosidase. This is the enzyme that will break lactose into glucose and galactose. And lactose permease will help to bring, or it will help to bring lactose from outside the cell to inside the cell. So, that is why the name is lactose permease. So, it will bring lactose molecule inside the cell. So, we are talking about bacterial cell here, particularly you may consider its E. coli cell. So, import of lactose. So, now naturally what will happen? So, if this is the gene, and in case of operon, in lac operon, all these three genes, LAC-Z, LAC-Y and LAC-A, they are controlled by single promoter, right? So, cluster of genes related by function. How they are related? All these three products of these three genes, they are helping in lactose metabolism. That is why related by function, and also regulated by single promoter and transcribed into one mRNA. So, what will happen? So, as you can see here, you have the promoter here. So, then as I already mentioned before during transcription lecture, that promoter will recruits this enzyme called RNA polymerase. So, then RNA polymerase will move in this direction and it will make mRNA, right. So, it will make mRNA, and then when RNA polymerase will move in direction, then you will get, because of the transcription. So, transcription, and you will get mRNA 5 prime to 3 prime, and this mRNA will be translated into this protein, because in this mRNA you have the information to make the LAC-Z product, LAC-Y product and LAC-A product. That means beta galactosidase, lactose permease and thiogalactoside transacetylase. So, now the thing is, although you can see here just one mRNA, but those three protein, they will not be fused together. They will be three separate protein, because those genes are separated by their own start codon and stop codon, and so that after the translation we will get three different protein like beta galactosidase, lactose permease and this protein thiogalactoside transacetylase. Now, the thing is, this is the normal scenario when the RNA polymerase bind to the promoter site, and then it will make the mRNA and it will make all the product. But the question here is, if lactose is not present there, in the environment where the bacteria is growing, then why the bacteria will make lactose? It is not required to make this lactose metabolizing gene product. Like if lactose is not there, there is no use of beta galactosidase or lactose permease. So as a result of that, the gene should be turned off, right? So as a result of that, I will discuss this part. So here, whatever I mentioned in the previous slide, that is the outcome of lac operon. If everything get transcribed, translated, then all these three products will be formed and they might help in lactose metabolism. Now, if lactose is not present, then bacteria will not make this this product. Those are unnecessary. So that means this operon is somehow regulated so that without requirement it will not make the product. How? So I am going to explain that. So although I mentioned lac Z, lac Y, lac A and promoter site, but I didn't mention this operator site. So you can see here it's mentioned O. So this means operator site present on DNA in this lac operon, operator site. So this operator site, in this place one protein will be present and it will bind here. So I would say this is a protein here. This is called lac repressor, lac repressor. It is a protein, it will bind to operator site. This is I would say it is like default state, like this lac repressor is always bound to the operator site, so that unnecessarily this product will not be produced. So now if the lac repressor is bound here, then this is our RNA polymerase. RNA polymerase, even if it get recruited in the promoter site, but it cannot transcribe these genes because lac repressor actually inhibiting the progression of RNA polymerase. So as a result of that, no expression of these genes. So now in this case, I am telling that no lactose here, lactose is not there. But now if lactose is present, then what will happen? If lactose is present, then see, in this case, I am saying that lactose is present. So if lactose is present, now from this lactose, like one isomer called allolactose should be there inside cell in a very, very low amount, allolactose. It's just an isomer of lactose. Now this allolactose will bind to this lac repressor. This is lac repressor. So now this allolactose, so if I say this is my allolactose, so allolactose will be binding to the lac repressor, and it will change the conformation of lac repressor so that it cannot bind to operator site anymore. So as a result of that, this operator site will be free. Now the RNA polymerase, so here this is again RNA polymerase. So now it can again move in this direction because the lac repressor is no longer bound to the operator site, because the allolactose bind to the lac repressor and it somehow changes its conformation so that it cannot recognize operator site anymore. So as a result of that, again the transcription will be happening here and you will get mRNA, and finally you will get protein required for lactose metabolism. So I just mentioned in presence and absence of lactose how it behaves, right? Now this is a very simplistic view of lac operon, but there are some more complexities there. I would like to mention that. So now bacteria, like if lactose is present and lactose is absent, I told. But what about if lactose and glucose both are present? Then like they evolve in such a way that like they will prefer glucose because they don't need to metabolize lactose anymore because ready source like glucose is available in media. So they will uptake, they will utilize glucose only, not the lactose. So as a result of that, again during that time they should like turn off their lag repressor, like not lag repressor, the lag operon. They have to turn it off, right? So that kind of mechanism is also there. So that is because I'm not going into like much more details, that is not required at this level. But the thing is, here you have another site that is called cap binding site. Cap binding site. This cap binding site means catabolite activator protein, CAP, catabolite activator protein binding site. Okay. So now the thing is, here one protein called catabolite activator protein will bind here, and it will actually, I would say this is some kind of glucose sensor, that how much glucose is present inside the cell. Depending on that it will bind and it will regulate this gene expression. The very briefly, if I mention, if huge amount of glucose present inside the cell, then the amount of cyclic AMP, so cyclic AMP should be low. So if cyclic AMP is low, then cyclic AMP will not bind to catabolite activator protein. So as a result of that, it will not actually turn on this system. But the glucose, if the amount of glucose is low inside the cell, then because of the metabolism, then we will have more amount of cyclic AMP. So this cyclic AMP actually bind to this catabolite activator protein, and after that it will actually help the this expression of this set of genes. So as a result of that, glucose and lactose both decide the fate of this lag operon, not just lactose. Okay, but finally they will use, I would say there are 4 different scenarios if I mention here now. Here glucose, lactose and lag operon, whether is on and off, that I will mention. For example, glucose is not there, lactose is not there, so I am just putting minus in both cases. Then why lag operon need to make this product? No lactose, so not required, right? So it should be off, turn off this whole system. And now you have you don't have glucose, but you have lactose. Then bacteria should depend on lactose for energy source, right? So as a result of that, they have to make this product from the lag operon so that they can metabolize lactose and they can survive. And I mentioned that if lactose is present, then how they regulate the gene expression here. So no glucose and then lactose is present, so this lag operon should be turned on. Now you have glucose but you don't have lactose, then there is no question of lag operon product, because no lactose present here. They can readily use glucose for their energy source. So this should be again off, but both present, then this is little bit complicated case, that lag operon, they shouldn't make product because they have glucose and they don't need to metabolize this lactose for their energy source. They can readily use glucose for this purpose. But the explanation relies on this position, that this glucose sensor catabolite activator protein, and because of high and low amount of glucose present inside the cell, that will dictate the expression of this this lag operon also, and that way these genes are regulated and they help in lactose metabolism. And because of this beautiful work, Jacob and Monod, they received Nobel Prize. I just mentioned in the previous slide, you can look into it. And so now this is just one example, and this is some kind of overview of gene expression. That's all for today, and you can look into any textbook, any standard textbooks will be fine for further reading.